Hi, my name is Aiya, and I'm going to be going over the experimental research design at a biological level. So basically, to properly define the experimental research design, it is when a researcher tries to draw a cause and effect relationship by comparing these two variables, which are going to be our independent variable and our dependent variable. So what goes down is the independent variable is manipulated in order to draw the relationship between that and the dependent variable, and the cause and effect relationship is hopefully established. So what researchers do in order to make sure that this is possible is they'll tend to make sure that the confounding variables are controlled, so they can make sure that they are properly deducing this cause and effect relationship. There are three types of experimental designs that are typically conducted, and I'm going to go over those pretty briefly. So our first one is going to be the laboratory experiment, and that is quite literally what its name is. It is a experiment conducted in a lab setting, and it is pretty credible. It's pretty easy to replicate. The one issue that stems from it is people tend to question how valid it is, because when you do a experiment in a lab setting, you can't really replicate it in real life. So people tend to question like how valid it is because it is done in an artificial environment. Second of all, we have the field study, which kind of helps with that issue in that it's done in a more natural setting. The independent variable is more natural. It has a higher ecological validity, but it is a lot lower in that you can't really replicate it. It's hard to replicate. And lastly, we have our quasi-experiment, which everything is natural. What happens in quasi-experiments are that it's super hard to replicate. Basically, researchers are waiting for this phenomenon to happen, and it's a lot of patience with these experiments. And so out of these types of experiments, I'm going to be going over a laboratory experiment conducted by Rozenzweig and Bennett, and these calico critters are going to be assisting. You know what would be so cool?
What?
What if we put rats into two different environments, like an impoverished environment and like a really nice environment, and we observed how it changed the chemical makeup of their brain, like the cerebral cortex. You know what? That does sound fun. Meet me in the lab tomorrow at like 7 a.m. We can get to work. So Rozenzweig and Bennett began their study, first looking into the impoverished condition, where each rat was placed in their own individual cage, ensuring isolation where no stimulus was applied whatsoever, as they were only given minimal food and water. The rats spent around 30 to 60 days in their respective environments before they were killed in order for the researchers to study the brain anatomy. In the other environment, the rats were placed in an enriched one where 10 to 12 rats were placed in a cage together, where they were provided with different stimulus objects to explore and play with. This group also received maze training and were given much better food and resources. They also dwelled in the setting for 30 to 60 days. However, they also ended up dying at the end. So, what did the researchers find when they were studying the impoverished rats and the enriched rats? What they found was the enriched rats had a thicker cerebral cortex, and they also had more acetylcholine receptors. And what this basically meant was that the enriched rats had it better when it came to thinking, decision making, and memory. And this basically meant that they had a better time learning. They were able to make decisions faster. Basically, having more stimulus made them smarter rats than the ones placed in a impoverished environment where they didn't have stimulus. And that is basically what the researchers found. So to link Rozenzweig and Bennett's study back to the original question as to why the experimental research design is used at a biological level of analysis, we can see that they were able to establish a cause and effect relationship, which isn't something you can usually do with other types of research methods, such as questionnaires. So basically, they manipulated their independent variable of environment, which was either the enriched environment or the impoverished environment, and they were able to gain more knowledge about their dependent variable, which was the neuroplasticity of the rat's brain. And the rats that were placed in this more enriched environment showed a better developed cerebral cortex. And so what we can do with this information is that we can generalize it onto humans, as animals and humans do share many similarities, and they were able to understand that just like the rats, people who are placed in better environments are able to have better developed brains, and this is a reason why the experimental research design is used at a biological level of analysis.